translation. So today we're going to start talking about transcription, which is the first part of protein synthesis. Okay, so up here, this is the central dogma of biology. So DNA provides the instructions for making protein. So this shows us how this happens. So DNA to RNA, okay, is how we go and take DNA from the nucleus, outside of the nucleus to the cytoplasm, to the ribosome in order to make a protein. So instead of going from DNA to DNA like we did in replication, we're going to go from DNA to RNA. So this is the process of copying DNA's instructions into RNA. So transcription. Okay, next week we're going to talk about RNA to a protein, which is the process of translation, which is the second piece to protein synthesis. Okay, so we've already talked about RNA, and we just reviewed this at the beginning of class. So ribonucleic acid is a long chain of nucleotides. Okay, we have ribose sugar, nitrogen or nitrogenous bases, AUCG, and it is a single-stranded molecule. And then this is the key that I've been telling you. Remember that RNA loves you. Okay, this is going to come into play today. So there are 3 types of RNA, and we have seen this earlier this week. So today we're actually going to get into those types of RNA. So the first one is mRNA, and this one is super, super important. Okay, mRNA stands for messenger. So this carries the message of DNA from the nucleus to the ribosome, which is found in the cytoplasm. Okay, so if you were to see a picture of mRNA, it would look very similar to this. Okay, it's a single strand as opposed to the double strand of DNA. So it looks very similar to DNA, but it's only one-stranded. Okay. The second type you see in the middle is rRNA, which stands for ribosomal. Essentially, this is what makes up the ribosome. That's it. Ribosomal RNA, ribosome. This one has 2 subunits, a large subunit and a small subunit. So you're going to see all of these types of RNA compiled together when we get into the translation piece. Okay. The last one is transfer RNA, tRNA, and this looks very much like a bubble tea. Okay. This is what carries the amino acid to the ribosome and binds with mRNA. So in your vocab, for those of us that completed it, on mRNA, you have what is known as the codon. On tRNA, you have what is known as the anticodon. So these are what match up. These are what match up in order for the amino acid to come in, the correct amino acid to come in. Because remember we said that the order of the bases is what determines the protein that's being made. mRNA. All right, so transcription, DNA to RNA. So DNA and the instructions that it contains is trapped inside of the nucleus because we've already talked about this. It is too large to get out. So DNA, because it is double-stranded, does not fit through the nuclear pores on the outside of the nucleus. Okay, so just like our pores on our face, too large, can't get out. So that is why the process of transcription happens, so that RNA, because it's single-stranded and is small enough, can squeeze through the pores of the nucleus. So DNA's instructions have to be copied into that smaller form. That smaller form is mRNA. Okay, so DNA to RNA, but very specifically mRNA is the process of transcription, okay? So remember I told you DNA polymerase we talked about with replication. Well, now comes RNA polymerase. And RNA polymerase is what breaks the hydrogen bonds in DNA. And it uses this as a template to be able to get mRNA. So very similarly to what helicase does for replication, RNA polymerase does during the process of transcription. Everybody follow? So mRNA leaves the nucleus with its message and it heads to the ribosome. So transcription takes place where? In the nucleus. It has to happen in the nucleus because what's there? DNA. And what did we say about DNA? Can't leave. It's too big. Okay, so transcription and replication both take place in the nucleus. Okay, so when we get into translation next week, translation is going to take place in the cytoplasm at the ribosome. Okay, so now we're going to get into some different base pairing rules. So the other day we talked about DNA base pair rules. A pairs to what? And C pairs to, and vice versa, right? So things are gonna start looking a little bit different. So do not get these confused. So RNA has uracil instead of thymine. Okay, so it's got that U instead of the T, which we already know. So therefore the base pair rules are gonna work a little bit differently. So if we have a strand of DNA, A is now going to pair to what? U, because this is RNA. C is still going to pair to, T is still going to pair to A, because the T is only found in the DNA, and then the C will still pair with G, okay? So C and G do not change. And if you're going from DNA to RNA, the T is still going to be here in DNA, so it'll still pair with the A. Your difference is going to be A pairing to U. And we're going to get lots of practice on this, okay? So what is the RNA message that's created from the following sequence? So this is going to work just like we did the other day. Instead of going from DNA to DNA, though, today we're going from DNA to RNA. So T would pair with what? A, U, nice job. C, G, A, T, T, G, A, awesome. So that is what your complementary strand would look like. We're just going from DNA to RNA now instead of DNA to DNA, okay? Let's try another one. A, which one? U, A, C, T, G, C, C, T, A, perfect, okay? So just make sure when you are given a question, okay, that you look what the question is asking for. Is it replicating DNA or is it transcribing? If it's replicating, we're going DNA to DNA. So the T doesn't disappear. But if we are going from DNA to RNA for transcription, that U is going to come into play. Okay? All right, let's go again. Awesome. We think we got the hang of it? Not too bad? All right, so now just like I had said the other day, if we were able to use whiteboards, this is what we would be doing. So this one's gonna be quick, we've only got 6. So make sure we look what the directions are asking for. What is it asking? Replicate, so what are we going from? DNA to DNA, okay? Ready? T. Perfect. Okay? Tap, tap. We're replicating. Perfect. What are we doing? We're transcribing. So now we're going from what? Perfect. Awesome job, guys. All right.
At this time, all sophomores F through Z that need to have makeup pictures, please go to the auditorium lobby. Sophomores F through Z.
Nice. Awesome. I like those. Those are easy. Yes. So tell me. We're going to what? Yes. Y'all are rock stars. Perfect. Boom. AUA, nice job. All right, everybody got the hang of it?